Great to be here. So as Lily said, I'm a research fellow at the University of Birmingham with Alan McNally, and I'm going to talk today about the evolution of multidrug resistance in E. coli and how I think that metabolism underpins everything. So we can think about the trajectories for multidrug resistance as sort of a simplified timeline, if you like. On the very far left, we have sensitive isolates, and these can acquire small genetic changes that mean they can become resistant to antibiotics. So things like GIRA and PAR-C that give you fluoroquinolone resistance. And then more towards the right-hand side of the spectrum, we have multidrug-resistant isolates. And this can be when strains acquire genes like extended spectrum beta-lactamases or carbapenamases. And these are often encoded on multidrug resistance plasmids that can be acquired by conjugation. And so I'm really interested in the whole of this trajectory, and I think metabolism is really important at all of these different stages. So the organism that we look at is E. coli, partly because it's a really important global pathogen. This plot may well be familiar to many of you. It shows the number of deaths that are either associated with or attributable to antimicrobial resistance. So you see on the very left-hand side of the plot, you have E. coli as a leading cause of these AMR-associated deaths. So in and of itself, it's a really important pathogen, but it's also really interesting from a fundamental biology perspective as well. It's a hugely diverse species. Lily's really kindly introduced it for me, which is great, so we can think about it in terms of phyla groups and sequence types, clades and clones. And what's quite interesting is that we have multidrug resistance in some of these lineages, but not in others. And this then raises the question, well, how does this occur and why? And it's not even just within these. We can see within lineages, we have these differences as well. So this is a tree of ST131, or the Roman Empire, I suppose. And we see it has a really distinctive three-clade structure. So at the top, we have clade A, which is our ancestral lineage. And here, we don't really see any multidrug resistance. Whereas at the bottom, we have clade C. And here, we have our global MDR clones. So we have fluoroquinolone resistance and the gain of genes like CTXM. And so we can ask, like, how does this happen? Why do we get these differences in multidrug resistance in what is essentially a single species? And there's lots of different questions that I'm really interested in asking about this. So on the kind of left-hand side of the spectrum, we can ask whether mutations in certain genes can potentiate a resistance phenotype. And this can be in genes that aren't necessarily directly involved in AMR, so things like metabolic genes. And this is some of the work that I'm doing at the moment, doing large-scale experimental evolution with Alan at Birmingham. And then on the other end of the spectrum, we can ask, what happens when E. coli acquires multidrug resistance? This is often coming in on plasmids. What happens in the immediate aftermath and then afterwards? And overall, we can ask, what drives the evolution of a multidrug resistance clone? And so it's this second part here that I'm going to talk to you about today, but I'm really interested in the first part, and if anyone wants to talk about it over lunch, I'll be really happy to. So we know that when plasmids come into species and strains, that we get a transcriptional response. But what we don't know is what the nature of that is. Is it plasmid-specific? So do you get the same transcriptional response regardless of the type of MDR gene, the type of plasmid, or the size of the plasmid? Or is it much more host-specific? And this is a really important question, and we don't exactly know the answer. We don't know if it's pathways that are being affected or if it's the same genes that are being differentially expressed regardless of the type of plasmid. So this is something that we were interested in looking at. And so the first thing that we needed to do was gather a diverse set of multidrug resistance plasmids to be able to investigate this. So these are the plasmids that we have collected. They are from clinical and traveller studies, both E. coli and Klebsiella. There are very different sizes, from 57,000 base pairs up to 160,000 base pairs. They have different plasmid types. The genes in orange here are showing that they are conjugative. These are all the TRA genes. And they have different MDR genes. We have like CTXM15, 55, 27. And we have an oxygene up in the top, and mobile colistin resistance as well. And they're not only different in terms of the MDR genes, they also have lots of different genes on them that are very different between them as well. So this is a pangenome of the plasmids that we've selected. The genes are coloured roughly by function. Lots of them are black, which suggests that they have unknown function. Lots of genes in plasmids are hypothetical. So these are our set of conjugative plasmids. We then introduce them all in biological triplicates into E. coli MG1655, which is just a standard lab strain of E. coli, and this means that we can control the genetic background. So we introduce them by conjugation and then measure the immediate transcriptional response by RNA-seq. And so if we were expecting to see an entirely host-specific response, we would expect to see the same sets of differentially expressed genes regardless of the plasmid type. These plots here show the number of differentially expressed genes across the 8 different transconjugants. So here we're seeing upregulated genes in orange and downregulated genes in blue. So we have different scales on each of them, but overall the general pattern of them is quite different, which suggests that we might be seeing a plasmid-specific transcriptional response as a result of acquiring MDR plasmids. I'm just going to zoom in on a couple of examples for you. So this one is looking at 2 different type plasmids. We've got an I type on the left and an F type on the right, but they both encode CTX755, although the one on the right also has a mobile colistin resistance gene as well. And what's immediately of note, in the plot on the right-hand side, we see this really large overexpression of INS-H, which is a transposase involved in the movement of IS elements. And that's only in one of these, but we do see these sorts of genes cropping up across the data set. In our plot on the left, we're seeing metabolic genes being hit. So in the downregulated genes, our most highly downregulated, we see GLUT-D and GLUT-A. These are aerobic and anaerobic glycerol 3-phosphate dehydrogenases that are involved in glycerophospholipid metabolism. We also see the methionine genes there as well. In the upregulated genes, we have carb, again, lots of metabolism-based genes here. In LA232, our one on the right, we see things related to transport and the membrane. So YNEM is an inner membrane protein. We have BAME, which is involved in putting beta-barrel proteins into the outer membrane. So on a kind of overall level, we're seeing transport and metabolism genes being hit. We also see this in these transconjugants as well. So these are 2, again, we have different CTXM genes, same plasmid type, roughly comparable sizes as well. Again, if you look at the genes in the top right-hand corner of either of them, we see transposases. And here we get lots more genes that are involved in transport. So on the left-hand side, we see that YNEM again, the inner membrane protein. And we also have a downregulation in LSR A and C, which are involved in the transport of autoinducer 2, which is involved in quorum sensing. And then on the right-hand side, we have our zinc transporters, ZINT, which is involved in the transport of zinc. So lots of transport, lots of metabolism genes that are being hit. So this suggests that perhaps what we might be seeing is, yes, very specific transcriptional responses per plasmid, but on the whole, metabolism and transport being really important. Now, I really love metabolism, so it's probably better to look at things in a slightly less biased way. So we did some go-enrichment analysis with the whole group of transconjugants. This heat map here, the darker blue, we're seeing a higher fold enrichment of these differentially expressed genes. And again, we're seeing metabolism and transport unambiguously differentially expressed across the different transconjugants. Now, I just want to pull out 2 specific examples for you now that we're seeing across the different plasmids. So the first one is related to methionine transport and metabolism. And this is a rough schematic of the pathway here for you. So where there's the dashed lines on the very left-hand side of the plot, I've kind of collapsed those reactions into one. So we're seeing the transfer from aspartate through homoserine into homocysteine. MET-E and MET-H are both converting homocysteine into L-methionine, but MET-E is cobalamin independent and MET-H is cobalamin dependent. And then we've got the regulator here as well, and then the transporter too. This is just to orient you. And then what we see across the transconjugants is a consistent downregulation of genes in this pathway, regardless of the type of plasmid. And so we see it's not occurring in all of the transconjugants, but when we see these being significantly hit, they are always being downregulated. And this is specifically and most strongly seen in MET-A and B, which is towards the left-hand side of this plot, converting homoserine into homocysteine, in the regulator and in MET-E as well. And so what this could mean, possibly a result of impacting translation, or this section of the reaction at the very end here on the right-hand side, converting methionine to S-adenosine methionine. This is really important for things like polyamine biosynthesis. So this could be another possible downstream reaction that could be important. The second example is looking at the significance of arginine transport and metabolism. So again, here we have some simplified plots to kind of illustrate the point. Here we have circles that show upregulated genes and squares that show genes that are downregulated, and they're coloured by the relevant transconjugant. So what we're seeing at the top, we have an upregulation of ART-J, which is involved in the import of arginine. And then at the same time, we're seeing an upregulation of genes that convert glutamate into arginine. So this suggests we could have a potential need for arginine as a result of these incoming plasmids. At the same time, we're seeing a downregulation of ARG-O, which is involved in arginine export, and a downregulation in the genes that convert arginine into either glutamate or into succinate for the TCA cycle. So there's kind of a couple of different possible explanations for this. One, we either have a requirement for arginine, or potentially what we're seeing is a diversion of resources away from the TCA cycle. And again, arginine is something that's really important in things like polyamine biosynthesis. So just to conclude, we can see that we have transcriptional responses to plasmid acquisition that do seem to be plasmid-specific. But what we see is metabolic genes commonly being reprogrammed across the different transconjugants. And we see this transcriptional response is not significantly associated with the size of the plasmid. And we've also done some competition assays to show it's not to do with the fitness cost either. So we do think that this could be a metabolic underpinning. We see a downregulation of genes involved in methionine transport, and then a possible requirement for arginine or a diversion of resources away from the TCA cycle as well. And so with that, I'd just like to acknowledge Alan McNally, who's my PI, and all the members of the McNally and Van Schaick lab groups, and did a lot of the help with all the sequencing, and we also did some of our sequencing through Microbes NG as well. And I was also funded by a college RDF award for the RNA-seq. So thank you very much.